In this video tutorial, we will explain how to perform a seeding experiment with the Crystalline instrument. Seeding is a commonly used tool in controlling the point of nucleation, polymorphic outcome, and influencing the final particle size distribution of a crystallization process. To perform a seeding experiment with the Crystalline, you will need the seeding assembly which is provided in the instrument starter kit. This includes the anti-solvent cap, a syringe, a tubing to insert the seeds, a connector and an adapter, and a tubing in 2 pieces with stoppers. First, click on New Crystalline Experiment and fill in the details, such as experiment name, username, and description. Select the anti-solvent cap to introduce seeds into your reactor later in the experiment. Fill in the vial details such as compound, solvent, concentration, and type of stirrer. Next, go to the Program tab to program your experiment. Drag and drop the experiment actions from the left side. First, add a linear ramp to dissolve your starting material. Set the end temperature above the solubility temperature. Then, add a prompt to ensure everything dissolves. Then, add another linear ramp to cool down to your seeding temperature. Add another prompt for the moment you will introduce your seeds. Finally, add a hold step to ensure that once the seeds are added, they will sustain and propagate. We recommend to add your seeds when the sample is slightly supersaturated, so they do not dissolve. The supersaturation and seeding temperature can be determined from the solubility and metastable curves phase diagram. You can learn more about how to determine these parameters by following the Crystalline or Crystal 16 video tutorials linked in the description. Next, in your lab, prepare to load your seeds. For this, you need to tap the end of the tube into the powder, similar to preparing a melting point capillary. This is best done with fine powder material, so you may need to gently grind the seeds using a pestle and mortar. This will create a small plug of seeds that can be pushed into the sample. Typically you will need a few centimeters of seeding in the tubing. Then, connect the syringe with the plunger pulled back to the tubing using the adapter. The air in the syringe will be used as a propeller to push the seeds into the sample. Finally, prepare your sample vial with the anti-solvent cap and connect the 2 short tubing pieces with the stoppers. Place the sample in the desired reactor outlet position. In the software, go to Run and Results tab. Select your reactor and start the experiment. When the experiment reaches the seed prompt action, you will see the seed prompt appear on the software screen. This is when you can pull the vial up briefly from the reactor to check it is at the correct height. Remove one of the tube stoppers and connect the tube to the syringe. Then press down on the syringe to push the seeds into the vial that is in the reactor. You can see the seeds enter and propagate in the reactor on the camera. Press Continue on the prompt to continue the experiment. You can explore your experiment either real-time or after it is completed. The graph shows the reactor temperature in red, the target temperature in orange, the stir speed in gray, and the transmissivity in light blue. Click on either of these parameters to see or hide them in the graph. Using the sliders at the bottom, you can zoom in the various parts of the graph. You can right-click and select data to see the captured image at that point in the experiment, and move to pin to search through the images. You can also search through the images using the arrows left and right. To reset the view of the graph, right-click and select Reset Zoom. Click on Export to export your experiment as CSV file, images, or movie. You can export the entire experiment or specify an interval, then click Save. To analyze the data, go to the Analyze tab. Please check the dedicated video tutorial on how to analyze particle size distribution using the Crystalline. You can find the link to it in the description. Finally, you can save the experiment by going to File and clicking Save and Close. Thank you for watching. If you have any questions, please reach out to us.